Okay, so welcome everyone again. This session has been organized by the BioClock Consortium, and the BioClock Consortium is an NWO funded association with researchers across the whole Netherlands who aim to restore and preserve the health of the biological clock, from human health to the natural environment and the protection of biodiversity. And within 3 clusters of this consortium, we aim to promote the relevance of the healthy biological clock in society, but also to improve patients' care using chronobiology in clinical practice, but also to develop strategies to minimize the negative effects of, for example, light pollution in the environment. And for more information and exciting news and ongoing projects, you're very welcome to check out our website, or to also follow us on social media to not miss anything. So, for example, LinkedIn and Twitter. Yes, Johanna.
Yeah, so hello, everyone. It's me, Johanna. So within the same frame, what Laura just said, we kicked off today the BioClock Academy. This is a lecture series which will take place every 3rd Wednesday of the month. It is addressed to anyone who is interested and new to the field of chronobiology, like, for example, the early career researchers. And its aim is to introduce you to the basic concepts, principles, and understanding of the biological rhythms on all its levels, what briefly Laura mentioned earlier, like in health, in society, and in the environment. So all topics which are covered by the research in the BioClock Consortium. Therefore, within the BioClock Academy, we invite every month experts from the field, which will explain the basic concepts of chronobiology and will hopefully pack you with and equip you with the proper knowledge in your upcoming studies. And yeah, the time frame today, we will have 40 minutes of the talk given by Johanna Meijer, and this will be followed by 15 minutes of discussion. So roughly 1 hour. We hope to finish at around 5. And during the talk, you can write your questions in the chat so that we can discuss them at the end of the talk. Yes, and the topic of today will address our central circadian clock and its neural organization. And the talk will be given by, like Professor Johanna Meijer. She's a neurophysiologist at the Leiden University Medical Center in Leiden. She is head of the Circadian Clock Lab within the Cell and Chemical Biology Department of the LUMC, and also one of the initiators of the BioClock Consortium. Her research focuses on the investigation of the neural networks of our internal biological clock, the suprachiasmatic nucleus. She therefore addresses exciting questions like how does light affect the SCN? How does this relate to entrainment and seasonal encoding of the biological clock, but also how we can strengthen the clock function. And she therefore applies a multidisciplinary approach by combining electrophysiological recordings and imaging techniques. And with this brief introduction, we want to give a warm welcome to Professor Johanna Meijer. We are very looking forward for your talk. And yes, the virtual stage is yours. Welcome.
Yeah, thank you, Laura and Georgiana. It's great to open this series of lectures now with this first very introductory lecture. And indeed, as was said, it is a lecture that is aimed also for educational purposes within the group and for new students who are entering the field. This will be very basic, and being the first one of the basic lectures, it is basic. Yet, since it is given from my perspective, it will be neurophysiology focused. So now I will start to share my screen, and I hope you can see my PowerPoint presentation. Okay. So there we go. Our internal clock, and now I will talk about the basic of biological clocks. Our internal clock, as we know, has evolved very early in evolution. So the very first single-cell organisms were already able to deal with the changing environment of the daily day and night cycle by their internal clocks. And for instance, single-cell algae living at the surface of the sea were able to predict the sunrise and were then able to move to the deeper layers of the sea to protect themselves against the UV radiation, which was harmful for them. So this internal mechanism, one important reason to have that is that you can anticipate changes in the environment. Now, this internal mechanism turned out to be so crucial and such a powerful mechanism to increase fitness of animals that after that it has been internally incorporated in all the organisms that have evolved. There's hardly any organism that we know that has no biological clock. So as I said, the day-night cycles are caused by the rotation of the Earth around the axis, leading to these day-night changes from the beginning of evolution onwards. In the course of evolution, the animals, of course, they evolved later in evolution, but these animals, rodents, insectivores, mammals, they specialized in being either capable of dealing well with the dark nights or dealing well with the bright days. And those animals turn out to be quite different in their organization of the circadian system. So for one reason, here is a picture of a tarsier. It has very big eyes. It is a nocturnal animal and is able to see very low light intensities still when it is dark. But many of the nocturnal animals have actually given up their visual system as a very important system and rely more on other senses. On the other hand, you see a squirrel on the right side. The squirrel is a typical diurnal animal. And if you look at the brain of diurnal animals and nocturnal animals, they are quite different because the brain of diurnal animals has very large areas that are devoted to visual tasks, very large areas. And they jump from branch to branch. And their circadian system has evolved in such a way that they are active during the day. So we can record the activity rhythms from animals, for instance, by giving them a running wheel, and many of the animals run in running wheels, even cats do and rabbits do, and I mean many types of animals actually run in wheels. But in laboratory settings, we record often hamsters, mice, transgenic mice, rats, etc. These are, of note, all nocturnal animals, and all these animals that are used in mammalian research on circadian rhythms have been mostly nocturnal. Now, if we look at the activity rhythm of such a nocturnal animal, we can see that here in the following graph. Here the days are plotted beneath one another, and this is a very typical way to present activity rhythms in this field. So the first 12 hours of the day, it is light. The second 12 hours of the day, it is dark. Do you see my arrow? Yeah, okay. And as you can see, this nocturnal animal, in this case it is a mouse, confines its activity mostly to the dark area, so the nocturnal animal. But if you look careful here, you can see that the activity of the animal actually starts a little bit before the onset of the darkness. Can you see that? So it clearly shows that a nocturnal animal is aware of the change that is about to come. It is anticipating the change to darkness. It's getting active just before dark starts. Then at day 20, it is indicated DD, and DD means in our field dark, dark. From that moment onwards, the animal is released in constant darkness. And what we see now is the rhythm of the animal without any correction from the external light-dark cycle. So of note, the animal is still rhythmic. That's what we should note first. The animal is still rhythmic, but the period is not exactly 24 hours anymore. The period is a little bit different from 24 hours. And in this case, the animal starts a little earlier each day. And that means that the period is a little bit shorter than 24 hours. For instance, 23.5 hours, 23.7 hours. It is often in that range. So the animal starts to be a little bit earlier every day. But I also like to ask your attention for the high precision of this rhythm, because you can really put a ruler along the onset lines of the animals, even when it is in constant darkness. Now, if we had to judge the time, and if we had not seen a watch or a clock for 24 hours, we wouldn't be able to judge it with a precision of, well, less than an hour. But at the same time, our bodily rhythms, driven by our internal clock, of which we are not consciously aware, have the high precision and are timed with a precision of, well, with a variability of 1 or 2 minutes from day to day. So this is also noteworthy. So the rhythms continue with constant darkness. The rhythms are apparently not generated by the light-dark cycle. They are entrained, synchronized by the light-dark cycle, and they are highly precise. That's what we learn already from this. So in around 1920, 1925, there was the first investigator, Johnson, and he already noticed that the rhythms of mice were continuing in constant conditions. And he concluded there must be an internal clock that drives these rhythms. Apparently, it's not defined or determined by the environment. Okay. So the continuation in constant environment means that the biological rhythms we are talking about are endogenously driven. Now in humans, we also have a clock. We also have rhythms that are driven endogenously. But what you can see here from human studies, and this is a study from Meijer from 1979, an early study, where students could be a subject in a study where they could not know the external time outside, and they were able to turn on and off the light by themselves. So what happened on the first day, you can see the darkness here means the activity of the students was still in the light period. The yellow was the sleep, that was in the dark. But now, if they don't see the environmental cycle, if they are not corrected for that, you can see that in this case, the students, this single subject, became active later and later each day. And this means that in this human subject, the free running period, the revolution time, is longer than 24 hours, and the human being starts a little bit later every day. Well, this is typical for humans. Humans tend to have rhythms that are longer than 24 hours. And this is what most of us, especially around the age between 15 and 30 years of age, then it is longest, especially note in the morning. In the morning, it's difficult to get up because in fact, your internal system is not completely ready for it. Yeah. So whereas the nocturnal animals has the tendency to become active a little bit too early, as I showed you in the figure, diurnal animals, not all of them, but humans and many others, have the tendency to are a little bit behind with it. And you see that in yourself in weekends, and every Monday morning we experience, in fact, a jet lag, a small jet lag, because we were free running in the weekend and we're a little bit out of phase. Now, in 1972, the location of the clock in humans was identified by two groups independently, by Bob Moore and Eichler, and by another group of Irv Zucker, 1972, and they used the fact that clocks are sensitive to light and darkness as a cue of where it could be. So they checked the areas along the visual pathways in the brain, and they lesioned them one after the other and found in that way where the clock is located. And it turns out that the clock is located, that's the first station where the optic nerve is projecting to the brain cells. So if we now look here at the optic nerves, here's the chiasm, then we see here just above the chiasm, a group of 10,000 neurons on each side. And these neurons are at the base of the hypothalamus, at the evolutionary oldest part of the brain, the cells that produce these rhythms of about 24 hours. And via the optic chiasm, they get the information on the environmental light-dark cycle and can be corrected if they are out of phase. Monday morning, they are corrected. So the clock of the brain in humans is crucial for a number of rhythms that we have. So it's activity of different brain nuclei. It's the cortisol levels, for instance, at peak in the morning. It's cognitive functions, it's performance or reaction time. And as an example, our performance during the night is about similar as our performance during the day after 2 alcoholic consumptions. So this is, of course, important for people who have to work at night, who have to do a surgery or have to put a plane on the ground. These rhythms in performance are not trivial for many jobs where you have to perform at a high level. Heartbeat, sleepiness, melatonin levels, temperature, liver and intestines, and also sensitivity to medication as a consequence of all these biological rhythms in our body are all subject to 24-hour rhythms. Now we can visualize the 24-hour rhythms in the biological clock by coupling a PER2 bioluminescence promoter to one of the clock genes. And here we have zoomed in on the clock in yellow here. In white, you see the optic chiasm. And if we now record the PER2 bioluminescence as an indication of the activity of one of the clock genes, about which we are going to talk later in this series, we can see that these individual cells are all oscillating. And I start a movie now, and we quickly go through 4 days. And of note, the tissue is bathed in a very constant medium. Nothing happens in the medium. And yet we see cycle 1, cycle 2, day 2, day 3, and day 4. The cells in the clock are all oscillating, and they peak with their bioluminescence expression during the day, and they have their trough during the night. So what we can measure from these cells of the clock is the gene expression, which I was just showing. But as a neurophysiologist, we are also interested in measuring the electrical or neuronal activity of the cells. And we can do that in brain slices that contain the clock. So here at the bottom, you see a brain slice. In the white, you see the optic chiasm. And we can measure at in the biological clock cells, and we can measure then the action potentials that are given off by the cells. We can also do a recording in vivo where the animals are implanted with a microelectrode aimed at the clock. And now, and this is very important, and we get back to this, the clock is an integral part of the brain. So the clock is now also communicating with other brain areas, right? So it is not only receiving light, it's also communicating with other brain areas, and it is also receiving possibly input from the animal behavior. We get back to that as well. And then finally, we can measure the animal's behavior by itself. So here we have 4 different levels of analysis. And this is crucial in brain research, that you realize that you can do investigations at different levels of analysis, which gives you different parts of the answer in which you're interested. So as a neurophysiology group, we do recordings. If you do intracellular recordings, you measure the difference in membrane potential between the inside and the outside of a cell, but you also damage the cell. And if you want to do 24-hour recordings, it's not so easy if you have impaled the small with an electrode. They lose their vitality after several minutes already. So what we do in our group is, for the cells less invasive, we put an electrode outside the cells, and we try to pick up the changes in the electrical field that occur as a consequence of the action potentials. So we have an indirect measure for the action potentials. This is different from an EEG. That is an even more zoomed out way to record activity. But this is a way to measure a population of action potentials and what it is reflecting. So when we do a recording with an implanted electrode in the clock, in this case from an animal that is walking through the cage, we can see that in black, from day to day, the electrical activity in the clock is clearly going up and down. And in red, you see the behavioral activity of the animal. And when the animal is active during the night, the clock is low in activity. And when the animal is quiet during the day, the clock is high in activity. Also for diurnal animals, the animals that are active during the day, the clock is low in activity during the night and high in activity during the day. That's the same, but their output signal is reversed. So in diurnal animals, when the clock is active, the brain is active. And in nocturnal animals, when the clock is active, the brain becomes inactive, as a most simplified version, but that's partly correct. So these are recordings from a population of cells, and we have also performed recordings of individual cells. When we performed recordings of individual cells, we have several criteria by which we determine that this is an individual cell. And here you can see left on the top, the light-dark cycle. And when it is dark, or it is a projected dark time, because this is in a brain slice, here is one cell that is active for almost 12 hours. But here is a cell that's active only during the second part of the day. Here is a cell that's active predominantly at the first part of the day. And here's a single cell that is active during the night. So what we see now is that individual cells are not all active at the same moment of the cycle. But what happens, the individual cells of the clock are active at slightly different phases, and together they determine the ensemble activity of the clock, and this is the signal that is given off to the remaining of the brain. Now, having said this, at this moment, I also would like to stress that for neurophysiologists, and maybe for everybody, but electrical signals are the language by which brain cells communicate with one another. I mean, they can give up they can produce protein, they can express different genes, they can have whatever chemical reactions, but to have an effect on another cell further down, they need to produce action potentials, and that is the signal for the other area that is perceived, right? So this is the signal from the brain, and not only are these action potentials the language in a human brain or in a mammalian brain, but also in snails, in reptiles, in all sort of primitive animals, the electrical activity is the language. So what we see now, therefore, is that so far the conclusion is that the SN clock is a multi-oscillator system. There are many oscillating neurons within this clock. But now I want to stress that the clock is not only important for daily rhythms, 24-hour clock, but it is also involved in seasonal rhythms. So, oh wait, now something goes wrong. Yeah, so while the Earth is rotating around its axis, leading to 24-hour rhythms, the Earth is also rotating around the Sun. And clearly this produces seasonal rhythms in the length of the day. These seasonal rhythms in the length of the day lead to seasonal responses from animals. And in this case, we have two hamsters. And the brown hamster is kept in a long day. The white hamster is kept in a short day, while both hamsters were kept at 21 degrees. But just the duration of the photoperiod, as we call it, the duration of the day length, is the cue for animals to become, to take their winter physiology or their summer physiology. And this is very important for the animals for when they become reproductive, so that all animals have their offspring in the spring. Yeah. So we investigated how these seasons are encoded for by the clock. And here at the upper panel, you see that the cells, when they are slightly out of phase, they produce an ensemble rhythm, which is normally given off when the animals are kept in a 12-hour light-dark cycle. But what we found when we kept animals in a long day, or actually questioned first, is maybe if we keep them on a long day, maybe the cells spread out more in phase and now encode the day length by the red arrow given here in the middle. So this is what we tested, and we provided animals with a day length that actually we have in the Netherlands. So we had animals, so this is what we have in the Netherlands from January to May, June to December again. We have in the winter 16 hours of darkness and 8 hours of light. In the summer, we have 8 hours of darkness and 16 hours of light. So this is what we gave to our animals. And then we measured the cells and their distribution. How what you can see here is that, and this is the figure you already saw, from an animal that was kept on a short day, the cells are active more or less during the day, more or less in phase, but not completely. While if we have the cells or we measure from the clock of an animal kept on long days, we see that here a cell is active at the end of the day, beginning of the day, middle of the night, end of the night. In other words, in a long day, the activity of the cells spread out in phase. And therefore, if we simulate now our findings, we find the following mechanism for recognizing the season. In a short day, the cells are active close to one another, leading to a representation of a short day. And in a long day, and the cells are active far from one another, leading to an internal representation of a long day. So beautifully, we have now a structure in the brain, tiny as it is, which is aware both of the rotation of the Earth around the axis and also of the rotation of the Earth around the sun. So a cosmic hero we have here. So far I talked about the first 3 points that I'm showing here. I will now go into the effects of light on clocks. And after that, much more briefly about the effects of behavioral activity on clocks and about the effect of age on clocks. And this will be dealt with much more extensively in future lectures. But what I find most important now is to go a little bit in more detail into the effects of light on clocks. So in mammals, the light information is perceived exclusively via the eye. So there are no other ways by which the clock is informed by light than through the eye, through the retina. In many animals, like birds and reptiles, there are also what we call extraretinal photoreceptors. Photoreceptors that are not in their eyes, but which are like immediately under the skull or in separate organs. This is not the case in mammals. Mammals perceive, the clock of mammals perceive light through the eyes. And then how do they respond to the light? Well, this is a figure showing how the electrical discharge in the clock is depending on the light intensity that is perceived. So we see here in figure A that if we present a light pulse of 5 minutes and it is only 0.515 lux, which is very low, 0.15 lux, the clock responds to it and the discharge rates go up. What we also see is that it is going up for the full duration of the light stimulus. This is not what you will see in many of the other visual areas in our brain. Most of our visual brain areas respond only at the onset and at the offset of light, but they cannot code for the full duration of the light stimulus. The only other brain area which can do so is the pretectum, and the pretectum is involved in regulating our pupil diameter And of course, your pupils have to be narrowed down or widened up for as long as the particular illumination is there. Okay, so the clock is also doing that. It is responding to light for as long as the light is there. Also, the day length is fully coded by the clock because the full length of the day leads to an activation of discharge rate in the clock. Now, if we give 2 lux, then the discharge rate goes up to a larger extent. You see it goes to a little bit higher level than with 0.15 lux. And with 140 lux, it goes up to an even higher level. However, this is more or less the working range of the SCN. These are more or less the light intensities that occur at dawn and dusk. So whereas from midnight to a sunny day, there are like about 9 logarithmic units of light intensity covered, the cells of the clock can only discriminate between is it night or is it day, and can only have a very small working range in their sigmoid curve between the intensities of dawn and dusk, which makes sense with their task. As I said, the electrical activity of the clock is elevated in the nocturnal animals for as long as the light stimulus is presented. So a few minutes, 5 minutes, and here half an hour. The discharge rate stays up. And then finally, we see here when we present light pulses at an intensity that is a little bit in between being perceived and not being perceived, there we can detect that the highest sensitivity of the clock exists at the middle of the night. So in the middle of the night, we see responses to light that at daytime are not fully perceived by the clock. The other question is, to what colors is the clock responding? And many of us know that melanopsin is a crucial photopigment that is sensitive to blue light. Melanopsin is contained in the retinal ganglion cells and are directly light responsive. Now here we are doing recordings from the mouse SCN, and we present the mouse with ultraviolet light because mice have an ultraviolet sensitive receptor in the eye, and they have also an ultraviolet or a UV transmittant lens, which we do not have. We give also blue light and we give also green light. And we do this here in wild type animals, and we do this also in animals that lack the melanopsin. These mice were given to us from Oxford University, from Russell Foster. And what we see is that the animals respond not only to blue light, but also to UV light and green light, and that in the absence of melanopsin, the cells still respond to these 3 light intensities. That does not mean that melanopsin plays no role, but it means that there is redundancy in the afferent input to the SCN. Melanopsin is important, but without melanopsin, the clock is also light important. So we should not overestimate the role of one versus the other. And then at some point we received mice that have only cones in the retina. We received those from Seymour Hatter. And if the mice had only cones, no more rhodopsin, because rods and cones are the classical photoreceptors, melanopsin is not. Now they only have cones, and we can see that they still respond to the UV light, to the blue light, and to the green light, but to a lesser extent. So with only cones, they respond to a lesser extent. But it does explain that even if mice have only cones, they can entrain to UV light and to a lesser extent to green light. In fact, the UV cone is very sensitive and provides important information to the clock. We recently followed up some of this research in humans and put them in a 7 Tesla scanner. And this is one of our electronic engineers. Our electronic institute developed light emitting glasses with gives off narrow band colors at green light, blue light, or red light to determine what the response is in the human clock. And what we found in these experiments, and it is now just published by Robin Schoonderwoord, is that the response to violet light is minimal and not statistically significant, but that the response to blue light, green light, and orange light, which is our red cone, do all exist. So here we see the response of the mean response of many human subjects together. We see that there is almost no response to violet light, but blue, green, and orange light all lead to light responses in the human clock. So in conclusion about the light sensitivity of the clock's light response depends on the intensity levels that we have. It discriminates between intensities below and above dawn and dusk intensities. It can function, it is elevated for as long as the pulse is given off, so it goes for the duration of the light. It is depending on the time at which the light is provided, and it is depending on different colors. And that leads us to also now looking at the functional response of rhythms to light presentation. And this I find always a very essential thing for all students, whatever they are working on, to understand, and that is the phase response curve. So here we have a schematical drawing of an activity of a nocturnal animal. It's active during the night. And here at day 18, we release it to constant darkness. We know it has a period then shorter than 24 hours. And now in panel B, we continue, but now we give a light pulse of about 20 minutes at the end of the second half of the activity period of the animal. And we can see that this light pulse leads to an earlier onset of the activity of the animal on the next day. From there onwards, it continues with the period it already had, be it 23.5 or 23.7 hours, but it continues. And now we give pulse at the beginning of the animal's activity phase at day 20. And we see now that this pulse leads to a later onset of activity on the next day. So a light at the beginning of the night, the subjective night of these animals, leads to a delay of the rhythm. And this is essential, the response, the functional response of the clock, not talking about the neuronal activity, but talking about the functional response in behavior, depends on the time of the day at which the light is perceived. At day, while the animal is still kept in constant darkness, we give here a light pulse at day 29 in the middle of the resting period. That would have been in the middle of the day, but it's constant darkness here. This leads not to a shift. Now we're going to summarize that in panel C. So the onset of activity we call time 12, and this is an arbitrary definition, but it is always called time 12. Time 12 is the onset of activity. If we give a light pulse briefly after the onset of activity, as on day 20, we see that the phase shift is a delay, depicted as a downward point, a negative phase shift. Whereas if we give a light pulse at the second part of the night, we see an advance of the rhythm, depicted here at, for instance, time 18 maximal, as an advance of the phase shift. Okay. Now this phase response curve, not exactly this phase response curve, but delays at the beginning of the night and advances at the second part of the night, exist in all organisms on Earth, diurnal and nocturnal. The ability to synchronize is always depending on delays of light at the beginning of the night, advances of light at the end of the night. And this is such a beautiful principle because it is so general. So what happens? Let's check again at the panel A. Between days 1 and 15, right? When the animal is synchronized. Suppose the animal would come out of its burrow a little bit too early, at time point 11, for instance. Then it would be subjected to light, right? To the evening light. And what is the evening light doing? We look on the right, day 20, it delays. So the next day, the animal will not come out of the burrow too early, but is corrected for and will come out of the burrow later. Crucial to survive, because otherwise it's eaten up by a predator. On the other hand, if the animal would have gone back to the burrow too late, this doesn't have that tendency because the period is shorter than 24 hours. But this is what we have, humans. Suppose the animal goes too late back to the burrow, then it sees the evening, it sees the morning light, and by seeing the morning light, the rhythm is shifted forward. So the next day, the animal will go to its burrow earlier. And now, importantly, do I have it again? Yeah. So what do we need in human beings? We need corrections, we need advances. And we get them in the morning, especially on this Monday morning, this terrible Monday morning. And then we see light, and this light leads to an advance of our rhythm, what we need. Yeah. So animals with a period shorter than 24 hours tend to synchronize on this evening light. Animals, humans with a period longer than 24 hours synchronize especially on this morning light. Yeah. So turn the lights on in the morning, students and evening people like me. Then I will very quickly go and just mention that apart from light input to the clock, also behavior itself is feeding back to the clock. And if an animal is active, we can measure that. So this is a cage in which we can measure an animal with an implanted electrode. And with the red circle, within the red circle, we can detect the animal's activity. And what we have seen here, if we print all the points in which the animal has been inactive in blue, but all the points in which the animal has been active in red, we can see that when an animal is active, the activity in the clock is elevated. Now, this is a recording in a diurnal animal. So what does it mean? It means that during the day, if we are active, our biological clock, which should be active, is further activated by our own behavioral activity. So our own behavioral activity during the day enhances the rhythm of our clock. Isn't that amazing? So our own behavior activity is in fact part of a feedback loop, and by being active during the day, we strengthen our clock. I skip this. And the other point I want to make here is, because it relates to my talk, but we will get back to this later sessions, is that in aging, the clock decreases in functionality, and older people often fall asleep during the day. And we wondered whether the decrease in clock function is maybe caused by a decrease in the synchronization among the clock neurons, like similar as we have in long days. And we tested that in old animals. And indeed, we found that in older animals, the cells within the clock become desynchronized. They are not so much active at the same phase anymore. So now the rhythm becomes very shallow. And you will get more about that. So the overall conclusions that I had from my talk is that the clock of the SCN is a multi-oscillator. It is involved in both daily and seasonal rhythms. It is entrained by light, and we can have a phase response curve. We can, as a summary of the phase shifting effect, and which is similar across all organisms. behavioral activity during the day enhances our clock function, and aging decreases our clock function because it desynchronizes the SCN. So I would like to conclude by 3 remarks. One is that the circadian system is coded for by clock genes at single cell level. These clock gene expressions lead to rhythms in a membrane potential by the cells so that they give off cycles and action potentials. These cells form networks, which can then code for the seasons. This SCN network is part of a larger network in the brain so that it can perceive light input and also behavioral input, right? And this behavioral input can strengthen the clock. So point 1 is this is a challenge for brain research, that we have to understand how to integrate the genetic findings with the cellular findings, with the network findings, with the behavioral studies. So this was point 1 I was just making. Point 2, from our research and from this research showing that light desynchronizes the clock, I would like to stress that light pollution is therefore also a threat for nocturnal animals, because this light, this seemingly long days, desynchronize the clock. The animals think it is summer, but it is winter. They can be prepared to reproduce, but it is winter. So this artificial light can now encode for a wrong time of the year, right? And it can also weaken clock function as an aging. This is also a point clearly investigated in this whole team, and Camille Spoelstra and others will talk more about this, about the light pollution. Point 3, and this relates very much to my talk, mind the network. And of course, in biological research, I think 95% of the money goes to molecular research, to genetic molecular research. But not everything is in the genes, guys. I mean, look here, Niels Bohr, and we all share with Niels Bohr that we have about 20,000 genes. The cucumber has many more genes, but what the cucumber lacks is this complex network, yeah? And then this is really my last slide. If we now look at what properties are now determined at single cell level and at network level, then we have to conclude that indeed rhythm generation is a single cell attribute from which you will hear more. But the synchronization among the cells, the robustness among the cells, the adaptation to the season, the precision, the phenomenon we see with aging, the influence of exercise on the clock, these are all network properties. So mind the network. And then I would like to thank all the participants from this group and the grant support and the collaborations that I have. This is my personal website. And of course, you all already have the BioClock Consortium website. Thank you.
Thank you very much, Joke.
Yes, I stopped sharing my screen now.
Perfect. Thank you for the exciting talk. I think Laura has the eyes on our chat, which seems to be empty. We have time for questions, actually. So in case you didn't type it, I guess, Laura, we can also open. You can unmute yourself and ask a question or raise the hands and we unmute you.
Yeah, go ahead. And there are no stupid questions.
Yes, Bert is raising his hand. Could you unmute yourself, Bert?
Yes. Sorry for that. Thank you very much for a very nice introductory talk on the subject. Very intriguing. An obvious question, I suppose, so not stupid maybe, but obvious, is why is the rhythm in the biological species not exactly the same as the physical rhythm the Earth has for a day and a season? Why is it slightly longer than 24 hours? Why is that?
Suppose the clock would have exactly the same period as the rotation of the Earth, then there is no necessity to have a adaptation or a correction if necessary. And then once you're off, you were off, which is too bad. Apparently, the evolution has chosen for a system in which we have rhythms that are slightly different from 24 hours, but fortunately, we have this correction factor, right? So, and even if the rotation of the Earth would change further than it did already, we still have this great ability, and we share that with all organisms, to have slight corrections for the deviation between the endogenous clock and the rotation of the Earth. I would also like to mention that if we are very active, or if animals are very active, their period also changes a little bit. So it is very important that we have this capacity to adapt. Yeah.
I see Ayano is raising her hand, or the group.
Yes. Do you want to go first? we both of us have a question. Hi, I'm Esther. I'm from Amsterdam. I was wondering. So you talked about the strengthening of the clock by activity in diurnal mammals, but also nocturnal animals. Do you think, and this was supposedly mostly about movement, so movement activity being awake. What do you think is the effect of other behavioral inputs other than movement in animals on the SCN, like food intake? Oh, you're on mute. Oh, you're still on mute.
That's a very good question, I said, to Esther from Amsterdam. We tested that in one study, and in that study we had a video camera attached to the cage. And we did not only record from the clock, but we also recorded the type of activity that the animal was exhibiting. And then we distinguished between 7 or 8 types of behavior. And it turned out that, in fact, all these behaviors had an effect. But the more the intense the behavior was, the stronger was the effect. Yeah.
Okay, thank you. And Ayana also has a question.
Yes, a great talk, Professor Meijer. It was really fascinating and informative. I have 2 questions. First is about your human study. So you have looked into the response of towards different colors of light, and the like a receptor for different colors of light, does it have like a different sensitivity depending on the color? Like you said, like blue, so ultraviolet in human was not really responding, but the rest of the color, they were responding, but the sensitivity differs depending on the time of the day, or?
Yeah, okay. In these human studies, we could not study the time of the day effect. So it may be there, but we don't know. The different colors are likely to have a different intensity response curve. For instance, melanopsin, which is primarily sensitive to blue light, is very insensitive, relative to the cones, which are more sensitive. So the cones code for the different colors for in case of vision, but they have their specific wavelength sensitivity. Rhodopsin, which is mostly responsive for green light, but in fact has a very broad band spectral sensitivity curve, is the most sensitive photopigment. So when light intensities are very low, we know that the clock can respond to that and that there are changes in period in the clock. But for these very low light intensity, it is probably the rhodopsin that has mediated that. And maybe in those cases, rhodopsin is the main pigment signaling to the clock. And in those cases, green light would be the most important. However, if the light intensity goes up, then also melanopsin plays a role, and also the cones play a role. So then, well, it's more harder to say. It will depend on the animal, but then blue and light is also coming in as an important color.
Thank you. Also, another real quick question. So I was actually recently reading your paper from 2012 about exercise impact on strengthening the SCN rhythm, which was in electro, you were recording the spikes, I think, exercise can...
Yeah, okay, the figure that I was showing from the diurnal animal is still unpublished. We just submitted that to Current Biology. It was a study we did in the Arvicanthus. The Arvicanthus is a diurnal rodent. Yeah, so it is not published yet. And you were asking for the reference or not?
No, actually, no, it was,
sorry.
But good to learn that the figure was from Arvicanthus. My question was about that. So later you went through real quick about behavior impact on SCN, a behavioral peripheral import to SCN. And I saw your paper about how exercise can strengthen the rhythm, in SCN.
Nocturnal animal.
Would you explain that as is it rather a neuronal import? Well, how would you explain? Or is it like a hormonal import that is taking a big role? Because you said something about serotonin. You mentioned.
Okay, so I would like to give a complete lecture on this and also the diurnal and the nocturnal animals and their effective behavior on the two and light on the two. Maybe I get the option in several years from now if we rotate. But so what we think is that the behavioral activity is sensed, or the information is transmitted to the IGL, the intergeniculate leaflet, and also to the raphe, where the serotonin is produced. And from the raphe nuclei and from the intergeniculate leaflet, there are projections to the clock. And we expect, for several reasons, that those are the projections that are activated by behavioral activity. Also, because if you give serotonin to the clock, or if you give neuropeptide Y to the clock, which is in the IGL projection, you get the same acute effects and the same phase shifts as you get with behavioral activity. So one, there would be an anatomical ground for this. And two, in a functional analysis, they do mimic the phase shifting effects of one another.
Thank you so much. It's really clear. Yeah, thank you.
Okay, thank you everyone for your questions and your discussion. Unfortunately, we're kind of running out of time, so I think it's good that we are closing the session now. But thank you everyone for attending and for your interest. And I see there have been people from all over the world. So there will be some more interesting sessions in a month, and we will continue the BioClock Academy sessions. So please join again. And thanks very much to Joke for giving this really interesting talk. And so for the BioClock trainees, we were planning to have, you could stay in the session if you want to, but I'm now going to stop the recording, and thank you everyone for being here.
Thanks for being here. Yeah.